Hello, everyone. My name is Heinl. I'm a Senior Field Application Specialist for SCIEX, and today I would like to present to you in my part of the CE Fundamentals webinar how the CE-SDS actually works and how to optimize and modify your CE-SDS size mapping of your protein analytes. My talk will be divided in a couple of subsections. So I would start with the basic, how actually does CE-SDS work? Then a couple of influences on the CE-SDS sample preparation, how you can actually modify the temperature and what the influence of the capillary temperature actually does to the CE-SDS separation. And last but not least, a small topic on the laser-induced fluorescence detection and how to improve your CE-SDS assay, moving to new modalities, moving to low concentrated samples, more dirty samples from your classic antibody workflow. So how does CE-SDS actually works? Before I get more into detail, the basic history of CE-SDS is actually the SDS, sodium dodecyl-sulfate polyacrylamide gel electrophoresis. So this classic slab gel analysis, which probably every biochemist did during their career. You have a solidified gel. It's made from an anionic, or includes an anionic detergent, which is the SDS. During the sample preparation beforehand, we also add an SDS to the protein mixture, which then forms a complex in a ratio of 1.4 to 1 of SDS to 1 grams of protein. And due to the properties of SDS, they will basically, SDS molecules, they will coat the protein, which I will also explain in the future further slides. Due to the similar size or hydrodynamic volume to charge ratio, we require basically a separation gel to separate all our polymers. And this is done by the SDS gel. The neat thing is, due to the different sizes then and the SDS complex, applying an electric current, we can separate those proteins based on their size. Not unfortunately, but to consider is that some post-translation modifications or general modifications of the protein vary the hydrodynamic size to charge ratio. For example, if you have a high degree of glycosylation, this introduces another charge to the molecule, which then migrates differently to a non-glycosylated protein of the same size. So comparing the slab gel analysis on the left-hand side and the SDS CGE, so capillary gel electrophoresis, basically get almost the same results. So the proteins are separated based on the charge to size ratio. Nevertheless, the SDS page, even though quite cheap and easy to make, it's manual, it's messy due to the potentially cancerogenous labeling agents, staining agents. It's semi-quantitative because you have to stain the gel, you have to scan the gel, and then you're only able to do the data analysis. The resolution is somehow poor depending on how you create your gel, and the precision, especially from operator to operator, from day to day, from gel to gel, is poor, which means you just get a rough idea of your analyte, and it's certainly not being able to be reproduced in a QC quality controlled environment to release a drug, a protein drug. So this is where the CGE-SDS comes in place. Because it's an automated system, because it's an instrument, you can easily automate it. So you remove a lot of variability from operator, from gel, from the consumables itself. It's quantitative. So by just simply integrating the peaks, you get some sort of information in terms of concentration, in terms of relative concentration, as well as absolute concentration. The resolution, which I will also explain later on in this presentation, can be optimized, but from the get-go, as we have optimized it already, it is rather high resolving. As you see in this slide, you have already a really good separation between the non-glycosylated heavy chain, which is peak number 2, from the heavy chain peak number 1, which is not really seen on the slab gel analysis. The precision from day to day, the reproducibility is quite high. We are talking about a 2 to 5% reproducibility in migration time, as well as in relative area. So it can easily be implemented, and it's already since 2006 implemented in the QC environment to release antibody drugs. But nowadays, by moving to other different modalities, nanobodies, virus proteins, other differently sized proteins, we can implement and use this technology to also improve the quality control of the new modality drugs. So what is CE actually? CE is quite simple, to be honest. You basically do a gel electrophoresis or an electrophoresis in general inside of a capillary. The capillary itself here displayed in orange, it's an open bore glass tube. We're looking into an inner diameter from 25 to 150 micrometer diameter. Nowadays, the standard, let's say, size is 50 micron diameter, so really, really small. The length varies between 30 and 100 centimeter, whereas the standard assay, if we're talking about CE-SDS, is the 30 centimeter capillary. Looking at the volume, we actually fill just 200-ish nanoliters of consumables gel buffer into the capillary, so the usage is quite low, which is really helpful in today's environment to reduce the cost of consumables and the reuse of potentially difficult to substances which are difficult for the environment. Furthermore, once you have the capillary, you basically just need 2 vials with a conductive liquid. This conductive liquid is also now filled into the capillary. Then you have to have a high voltage power supply, which is then connected to the both reservoirs. And if you turn on the voltage, the charged molecules will start to move. Positively charged ions will move to the negative side, and negatively charged ions will move to the positive side. Due to difference in charge to size ratio, we get a separation, and then we can detect them at the detector. It sounds easy to build. It's actually easy to build a CE which does some sort of separation. Nevertheless, to have a capillary electrophoresis instrument which actually is reproducible, easy to service, easy to clean, and to be implemented in the quality control, it's much more challenging. And this is where we come in place with our CE portfolio, namely the P800 Plus, the BioPhase 8800 system, which is our new 8 capillary system as well, and which will be also heard in future talks, new device which is a chip-based CE system, which does have capillary isoelectric focusing directly coupled to mass spectrometry. So the CE-SDS in principle consists of a couple of different steps to be able to actually separate proteins with different sizes. So usually a protein itself, if you are in a solution, it's kind of this messy, crumbled up length of chain, a chain of amino acids. So you have all these tertiary, secondary, and quaternary structures. Nevertheless, if we're looking into gel, we actually need to somehow elongate, denature, untangle this protein mess. And this is usually done by heat. So during sample preparation, you have to optimize the heat to not destroy, degrade your protein, but to perfectly entangle the protein. The goal, or the most optimum case, would be a really long linear spaghetti, because then you have the proper migration through the sieving matrix. We also aid the unfolding entanglement as well as the separation by adding SDS in a ratio of 1 gram of protein to 1.4 grams of SDS. Due to this constant ratio, which we can actually achieve, we have a uniform negative charge on the outside of our proteins. Therefore, independent, and this is really beautiful, independent on the size of the protein, all of them have the same surface charge. What does it what is the outcome now? The neat thing is CE always separates based on charge to size. If you can exclude the charge out of this equation, the only separation background now is the actual size of its molecule. So in the CE-SDS, we can actually separate small-sized molecules from the larger-sized molecules, namely protein fragments to the intact protein. If we now have this all done in a capillary, we have a really neat and reproducible application to separate our molecules. So putting it all together, we have a capillary filled with a gel matrix. We have injected our samples, so the denatured proteins. We add high voltage to the mixture, and this means the small-sized proteins will migrate first, as indicated in this picture, and the large-sized protein migrates last. So you have a really neat separation, and then since you have the peaks, you can do all sorts of post-data analysis to determine purity, to determine concentration, and stability of your sample. So this application is already on the market since roughly 2006, 2009-ish, and it's widely applied for the IgG purity analysis of monoclonal antibodies. On the left-hand side, you see an example of 2 electropherograms. Once it's an antibody in the reduced condition, so by breaking off all the disulfide bonds, we receive the light chain, the non-glycosylated heavy chain, and the glycosylated heavy chain to determine deglycosylation, but also heavy chain to light chain ratios. In the non-reduced mode, basically keeping the antibody intact, we can determine whether our protein is stable in the chosen formulation buffer, in the chosen drug substance in conditions, but also in the formulated drug. Also, over time, you can do all sorts of purity analysis to see if your drug is still the drug you produced on first place. This is all at the end, basically, once you produce your antibody. Nevertheless, you can also apply the CE-SDS in other parts of the bioprocessing. Once you have the cell expansion, you can already check if your protein is there. During process development, you can check whether your purification steps would work in a certain yield, and so on. So it's a really universally applicable application. So I hope you learned a little bit now about the background of the CE-SDS. It's quite simple to understand. Now I would like to give you some outlook into how you can impact or how the sample preparation influences the CE-SDS separation. I don't highlight everything. I just want to point out certain things. One thing is really understanding the role of the SDS. The SDS provides the negative charge to the protein. So whenever you have something else in, it changes the size and therefore the separation properties. Furthermore, you really have to have this magic, let's say 1.4 to SDS to 1 gram of protein ratio. If you go underneath this ratio, you don't have the uniformly charged to each protein. Therefore, you don't separate only based on size. You would also then separate based on charge, which changes the separation profile drastically. Nevertheless, for certain proteins, it sometimes doesn't really work with applying or binding the protein if they have really poor denaturing, so if they don't really unfold, or if the SDS binding is somehow hindered to some post-translational modification. So how could you actually improve or optimize your denaturing or SDS binding? One point is really changing the denaturing temperature. Going higher, going lower, not only helps it with denaturing and unfolding, but also helps with SDS binding. Increasing denaturing time or modifying the denaturing time also helps to stabilize your assay. Thinking about having really short denaturing times might produce a huge variability if you have to produce a lot of samples. So usually a lower temperature with a higher degree of denaturing time helps with the reproducibility. Furthermore, some proteins are prone to degrade in certain pH. So what would be the worst case scenario if you destroy your protein during sample preparation if you want to see the stability of your protein? So sometimes you have to change the buffer, SDS buffer pH, We as CIEX, we will help you for this. We have currently 3 SDS buffers available. We have a TRIS HCl pH buffer, a pH 9 SDS buffer. We have a TRIS HCl pH 6.8 SDS buffer, as well as a 40 millimole phosphate SDS buffer at a pH of 6.5. Nevertheless, you're free to explore other pHs like a citrate MES buffer, phosphate buffer with an SDS content to help with your specific protein. If nothing helps, if it's a really complicated protein, you can also increase the concentration of SDS, so that helps to maintain the 1.4 to 1 gram of protein ratio to SDS to protein ratio. Another really elegant way also, if it's a really complicated, really large molecule, you can increase the chain length of the surfactants. So moving from sodium dodecyl sulfate to sodium hexadecyl sulfate. So this is a much, much longer chain. And this was already published in 2018 to help with certain size of fusion proteins with the unfolding, with the denaturing, with the separation itself. Another really important factor to see, and this is not really applicable for the sample preparation itself, but it's really applicable for all CE applications. In capillary electrophoresis, during a sequence, you will always have migration time shifts to longer migration time. If people are more known to LC, this is unusual, and you would say, well, this is something of a bad result. In CE, this is an intrinsic property. The reason is on capillary electrophoresis, all consumables are exposed to air. The caps we use to seal the vials are open to the environment, which means you always have evaporation of liquid from those vials. CE theory dictates the viscosity of the solvent you have for separation, determines or influences the speed of the molecule. So the logical turnaround here is if you have your solvent and you remove liquid from it by evaporation, you increase the viscosity. If you increase the viscosity, the molecules will migrate slower, which means the migration time of each individual molecule will increase. So we see it on the left-hand side. During a sequence of 24 injections, in our case 24 hours, we have a couple of minutes of migration time shift of each individual peak. Nevertheless, the purity, the reproducibility of relative peak areas still maintains the same, even though you have this migration time shift. If you want to remove or reduce the migration time shift, you can actually add mineral oil on top of your buffer refilling valves in your buffer tray layout, which then prevents liquid from being evaporated, and therefore this viscosity change doesn't happen, and you have perfectly reproducible migration times as well. So this is a neat procedure to help with migration time stability and helping with data analysis, certainly. Another factor is actually the harmful substances we use in capillary electrophoresis. The standard assay for the CE-SDS in reduced condition is beta-mercaptadinol to break off the disulfide bonds. Due to, well, the health concerns, but also the environmental concerns, it smells really bad. People were moving more to less harmful, less smelly substances. One of them, and usually used in gel electrophoresis, is DTT. Unfortunately, the DTT produces some sort of complex with our gel matrix, and therefore has a interfering peak with the non-glycosylated heavy chain and the heavy chain. If you have an interference peak here, you can't really use this analysis for protein size ratios. Nevertheless, and this we have published this year already, by adding iodoacetamide at least in a molar ratio of 1 to 2 of DTT to iodoacetamide and keep this mixture for 30 minutes in the dark, you basically reduce the DTT or alkylate the DTT with iodoacetamide, and then you'll remove this complex. So this is a really neat way to move from this smelly beta-mercaptadinol, potentially dangerous beta-mercaptadinol, to a less dangerous substance like DTT by just adding the iodoacetamide and waiting a little bit longer to remove, letting the complex being removed. So these were some highlights on our recent publications as well as to the influences on the sample preparation. Another thing is, well, resolution is always of a concern. Our assay is optimized for monoclonal antibodies, but what will happen if you move to different modalities? Resolution, you want to separate similarly sized proteins, so how you can actually influence and optimize the resolution. There are three major ways to optimize, and putting this all together actually achieves a really, really neat application. One thing is the change of separation temperature, so the capillary temperature itself. You can increase or decrease the electric field strength by increasing capillary length, by decreasing the voltage, and you can actually manage your injection conditions. And the last point actually ties into my last topic of today's talk about CE-SDS live, but more to this later on. So one thing is by changing the capillary temperature. You'll see these are two different gel matrices. One is dextran, one is PU, and you can basically see by increasing the temperature, not only the separation time will decrease, but also the peak shape will improve drastically. If we're looking at dextran, like the peak number 5 and 6, they're not well separated at 20 degrees Celsius, but by increasing the capillary temperature, we get a nicely separated peak 5 and 6, which is due to the different movement properties of those molecules at different temperatures, as well as the different viscosity of the gel based on the different temperatures. This is another example. By increasing or decreasing the capillary temperature, we can achieve a faster but potentially more resolved separation. And on this example of our SDS molecular weight size ladder. Looking at the marker resolution, actually, we can basically choose what we would like to see. Looking at low temperature over the course of all different sized molecular peaks, we have a beautiful separation and good resolution. Increasing the temperature, as we've seen in the previous slide, helps with the separation speed, but we decrease the resolution. So you're able to pick and choose what you would like to have, great resolution or high speed. Theoretically speaking, this can easily be applied for all sorts of molecules, so from small 10 kilodalton proteins to nanobodies to light chain to heavy chain and even larger ones. So the trend is always, if you increase temperature, the speed will increase, but theoretically speaking, the resolution will drop. Interestingly enough, this is not always the case for all sized molecules. So looking at this slide here, we see a drop of the resolution for the light chain. We see a drop of resolution for the heavy chain. Nevertheless, even though we expect a linear change, it's not linear. So you can really optimize based on your molecule of interest and the capillary temperature, what you would like to achieve. Furthermore, if we're looking at the nanobody, actually by increasing temperature, we increase the resolution. So this is contradictory to what we have seen beforehand. So really, if you do your method optimization, don't only look at higher temperature, also go lower in capillary temperature, which we can easily achieve by our liquid cooling system, which produces a really stable temperature to help with optimizing your assay. So my last topic for today is how you can actually use laser-induced fluorescence to help and optimize the CE-SDS assay. So laser-induced fluorescence, for the ones who don't know, it's a different technology to detect your analytes. Usually for standard, we use absorbance technologies, which means we shine UV light of some sort of wavelengths onto our capillary and measure the difference between the in light intensity when there's no analyte present and when there is an analyte present. The advantage of UV detection is whenever there is a substance which can absorb the light, you get a peak in electropherogram. The disadvantage of UV detection, whenever there is something which absorbs UV light, you get a peak in your electropherogram. So potentially buffer components, and other impurities can influence your separation profile, which means you might need to do some elaborate purification protocol to only get the protein peaks of interest. Laser-induced fluorescence, on the other hand, is a really elegant detection technology. We use a laser with a specific emission excitation wavelength. With this excitation wavelength, we excite a certain part of the molecule. Usually, the protein gets stereotized. This part then gets excited, and once it's released, it's emit a certain wavelength, light of a certain wavelength, which can be detected. So in this case, only molecules which can carry this stereotizing agent, this dye, will produce a peak. So you can clean up your electropherogram quite easily without doing an elaborate sample cleanup process. Furthermore, since we improve due to this specific nature of laser-induced fluorescence detection, we have a much, much higher sensitivity compared to the UV detection. So nevertheless, what do we also see about resolution? And last but not least, in today's environment, can we actually optimize everything to even get a higher throughput while maintaining high resolution, while improving the sensitivity of our analyte? So before I dive in on how to optimize, there are traditional 3 different dyes used in capillary electrophoresis or protein analysis for lift derivatization. The standard dye, and please excuse me, I don't want to read this out loud. In short, it's the FQ dye. It's already implemented in the industry, so it's a historic dye. Nevertheless, it does require some buffer exchange to begin with to work perfectly fine. This is some sort of labor-intensive process. Furthermore, and this is usually the most concerning factor, is the potassium cyanide used during the labeling procedure. If this workflow is already implemented, you probably don't have any issues with your EHS department. But if you want to implement this workflow nowadays in your lab, potassium cyanide is a red flag for the EHS department, so you probably not be able to implement this workflow. Due to all these sorts of buffer exchange, labeling reactions, and so on, we talk about roughly 45 to 60 minutes of sample preparation. To overcome the issue of the potassium cyanide, there is a dye called Tamra, which has been introduced a couple of decades ago. This obviously doesn't have the potassium cyanide. Nevertheless, you have to do a buffer exchange to start with, because this dye only works perfectly fine in a bicarbonate buffer. Furthermore, after the labeling is finished, you also have to remove the dye via a buffer exchange or removal process. So this increases the sample preparation time tremendously. In this case, we're talking about 70 to 90 minutes per sample. If you have a robot, for example, the Beckman Biomec i7, it's easy, but if you do this manually, it might take a while for a lot of your samples. This is where the last dye comes in place. It's the so-called Chromio P503. It's a beryllium dye which binds to primary amine. So whenever you have a primary amine in your protein, you're able to do this labeling reaction. The labeling reaction works in basically just the basic range of the buffer range, basic buffer range, which means if your formulation buffer is above pH 7, you're able to label your protein. Another really beautiful thing about the P503, the first steps of your reaction is just your simple, well-known and validated protocol for the UV sample preparation for the CE-SDS. You have your protein, you have an SDS containing sample buffer, you add reducing or alkylating agent to it, you denature the protein by heat, and you're done. In addition, you add then, after this denaturing step, you pipette in your dye. You heat it for another couple of minutes, 20 minutes, which certainly needs to be optimized. Let your sample cool down, dilute, and it's ready for analysis, which means no buffer exchange is needed. There's no potassium cyanide involved. You don't need to remove the dye after labeling reaction. the whole separation preparation principle is easy to automate because it only involves pipetting, mixing, and heating steps. So it's a really neat and quick workflow. So we're talking, depending on the heating times, of roughly 25 minutes of preparation time. Comparing now PDA or UV absorbance detection with the lift detection, you immediately see we get rid of all the potentially interfering system peaks, because those are just substances which absorb UV light. They're not getting labeled by the dye. You get similar profiles from the PDA to the lift detection. Please be aware this is a normalized trace view because lift is much, much more sensitive compared to PDA. Nevertheless, you will reduce, due to the addition of the dye molecules, the resolution. So this is one minor disadvantage moving to laser-induced fluorescence. Nevertheless, we can apply some CE theory to actually improve the resolution, help with the throughput, and to make CE-SDS live a really beneficial detection technology compared to your CE-SDS UV separation. So first of all, looking at the concentration, you'll see if we'll just look at the main peak from 100 microgram down to 1 microgram per milliliter, it's 100 to 1,000-fold less that we would actually have in your CE-SDS UV sample. So it only affects the peak height, but doesn't really affect the resolution. So it's a really reproducible assay independent of the sample concentration. Furthermore, since we have much, much higher sensitivity, we can actually reduce the amount which we will load onto the capillary. In this experiment, we varied the injection time as well as the pressure. So going from 5 seconds at 2.5 psi to 10 seconds, it doesn't really change the resolution. But the more we load, 10 seconds at 5 psi, and even up to 100 seconds at 5 psi, we drastically reduce the resolution. So by just loading less, we can, if we look at it from the other side, from high to low, we improve the resolution quite easily. And this is a really huge benefit about CE-SDS lift, because the sensitivity is higher and therefore you can add less or load less. Another thing which influences the resolution is the capillary length. In this experiment, we varied the capillary length from 10, 20 to 40 centimeter effective length using CE-SDS, using lift detection. And in this experiment, we actually kept the electric field strength the same over all capillary length to get rid of this factor. Therefore, looking at furthermore, so as we see, and this is based on CE theory, we expect improved resolution by having the molecules on longer capillaries, and you see it on the left-hand side, which dramatically improves the resolution between the non-glycosylated intact IgG and the intact IgG. Another variability, which I have mentioned before, is the capillary temperature. As indicated beforehand, we can actually improve the resolution also by having we drop slightly in resolution by increasing the capillary temperature, but by increasing the capillary length, we can also optimize the resolution as well. So what does it actually help? Well, we can actually put things together like capillary temperature, like capillary length, including laser-induced fluorescence detection, to put everything together to make a faster, similar resolving technology with lesser sample loading. So in this case, we compare a 20 centimeter CE-SDS UV separation in the yellow trace and the same molecule, but lower concentrated in the CE-SDS lift, but now at 10 centimeter with a different capillary temperature. And as you see, based on the resolution, we have achieved the same, but with a higher throughput. So in day-to-day environment where you have to get the result quicker and quicker, you can actually improve your throughput by just moving to laser-induced fluorescence and optimizing the other separation separation principles. So another and difficult to quantify advantage of using CE-SDS live is actually we can load much, much less. So even though you have your protein a high concentration, for new modalities or virus proteins, they actually are in high concentrated salt buffers. And for CE-UV, you might need to remove this salt to not influence the separation and the loading of the sample. On CE-SDS, live, on the other hand, we can use much lower concentrated samples. So by just simply diluting the sample, we can remove or reduce the salt, which has less influence on the separation itself and therefore better separation. Furthermore, and we can also, since we load less protein, the contamination of the capillary and the components is much, much less compared to the CE-SDS UV assay, which means if we load less, we can reduce the calibration times and cleaning times for the acidic wash, the basic wash, the capillary refilling. So this means you can improve the throughput even further by just removing and reducing those, by just moving to CE-SDS live. So I hope you enjoyed the talk, and we will have later on a Q&A session, so please keep the questions coming. In my talk today, I hope I was able to explain that the good sample preparation procedure is crucial for providing the optimum separation. If you adjust your temperature, and this is an easy way to improve your CE-SDS separation with our system, if you want to try out live, we strongly recommend using the Chromeo dye because it's easy to use and doesn't really impact the protein profile. Another factor to improve is the separation voltage. So by decreasing the voltage, you make things faster or slower by maintaining or optimizing your resolution. And last but not least, putting everything together also reducing the injection amount impacts the resolution, which means it does help with your analysis. So thank you for your kind attention, and I hope you enjoyed the talk. I'm here for your questions, and please enjoy the talks from my colleagues. Thank you very much.